Thank you, Simon. Thank you to the Black Sun family for sponsoring this lecture. And I apologize again about my voice, but I'm really happy to be here. This is my last of 6 lectures that I've given in the UK this week. We're going home tomorrow morning, and it's going to be really sad to leave this beautiful country, so I hope to be back and share more of my love for honeybees and pollinators with you in the future. So this has been a wonderful experience, and I really want to thank Simon Croson for inviting me. We met, if you weren't here in the previous days, we met last year in Florida, and I just feel very honored that he remembered me and that he invited me to come over to the UK for the first time, so thank you. When I offered the possibility to give a lecture about the biology of the colony, I think I was a little too overambitious, because you could probably take a whole semester of lectures once a week for 1 or 2 hours to go over topics about honeybee biology and the biology of the colony. So I've just chosen a couple of topics that are just biology-based of honeybees. One is going to be aging. So we're going to discuss a little bit how aging works in honeybees, just in case you might not know what's going on inside bees that are becoming older. Then I'm going to talk about foraging and the different aspects of bee math, so all the things that we have been able to quantify in the scientific world regarding foraging and the types of foraging available. And as promised, I have taken other stuff out so I can talk about the project with feral honeybees in South Texas and the work we've done with Nosema, because I promised that I would give that lecture today. So I'm going to briefly talk, well, for the last third of the lecture, or 20 minutes of the lecture, I will be talking about the work that we're doing with Nosema, Apis cerana in feral Africanized honeybees in South Texas. So they don't really all go together, these 3 topics. I apologize. I was just choosing some of the many, like the myriad of topics that we could go over for this lecture. So let's just dwell into aging for now. There is genetic and environmental plasticity in aging in honeybees. Most of you know, since you live here in the UK and you have a true winter, that summer bees or spring bees will live approximately 30 days post-emergence. But the in-hive bees can live much longer if you are able to prevent their foraging onset age. They can live quite longer, which tells us as scientists that foraging is a very demanding task that takes away basically the living of the honeybees, and they basically become much more frail and weak by doing all of the very demanding foraging tasks. In the winter, however, bees can live anywhere from 150 to 170 days on average. It has been recorded that they can live up to 350 days, although that's almost a year, so you would have to keep them in the hive without basically the ability to come out. But if you lower the temperature enough so that it's not the temperature proper to go out and foraging, you could keep bees living for that long. Again, this goes to show that foraging itself, being outside, is very energetically and metabolically demanding, and that's what's going to curtail the life of a worker. Typically, queens, we know, live anywhere from a year to 4 or plus years. Nowadays, we have this problem with queen supersedure, and so workers are quickly replacing these queens, and we no longer see a lot of queen breeders in the United States, at least, that can claim that their queens can live more than 2 years, really. I have a friend who's a very good queen breeder in the very high altitudes of Truchas, New Mexico, and she has queens that live anywhere from 4 to 5 years, and she breeds from those queens because she calls them survivor stock, and she hopes that those genes that make them hardy and long-living can be passed on to next generations, but typically nowadays queens only live 1 to 2 years. And they are typically killed prior to their very bad senescence or running out of sperm, like we've been talking about with queens of low reproductive quality. What are the symptoms of aging in honeybees? The major mortality of workers is due to metabolic damage from intense activity, again, typically foraging activity, that causes oxidative stress, just like it happens also in us. Nutritional stress, depending on how nutritious the food is or not. Aging causes malfunctioning in the central nervous system. And also it causes a compromise of the immunoresponses and the immune system. It causes an increase in the levels of juvenile hormone and stress-related hormones, and those things will lead a bee to senesce, because as we will see, juvenile hormone is only dependent on age mostly. What regulates aging in honeybees? Here we have a picture of a diagram of what would be a nurse bee compared to a forager bee. A lot of work on the production of vitellogenin, which is the egg yolk protein, and juvenile hormone has been done by Robert Page and his collaborators. He's out of the University of Arizona, and he has done a lot of work on the fluctuating levels of vitellogenin and juvenile hormone as they relate to aging. Interestingly, I've been talking about pheromonal communication quite a bit. We've talked about the potential odors that might be released by bees that are preparing to swarm. I talked in depth to you and also the other beekeeper clubs about queen mandibular pheromone and queen released pheromones that help with that decentralized control of task allocation in the hive. And not surprisingly, pheromonal contact between nurse bees, which are the young ones that feed the brood, and foragers, which are the old ones that go and get food, delays the transition to the foraging state. So something that is good for us to remember is that young workers, the nurses, have very large amounts of vitellogenin. And vitellogenin is the precursor for the brood food components. So very important hormone that helps in the production of brood food by nurses. And interestingly, juvenile hormone blocks the signaling of, vitellogenin blocks the production of the juvenile hormone. Again, nurses have very high titers of vitellogenin, which is always shown as a capital V, small g, and JH as juvenile hormone is higher in the foragers. Stress, which comes from physical disturbance, isolation, disease, starvation, or the onset of the foraging age, will increase the production or signaling of juvenile hormone, which leads to behavioral changes, which is the foraging age onset. So a lot of things are happening inside the brain of a nurse bee, environmental factors as well as physiological factors that will then lead to a reduction in the production of vitellogenin and then increase in the production of juvenile hormone, which is directly related to the onset of foraging. Overaged nurses, those that have typically longer lives than typical workers, so imagine that a typical worker lives about 30 days. If you're able to manage to keep bees that are overaged, for instance, if you preclude them from foraging, they are still able to produce brood food. So there's plasticity, and I've talked about plasticity which is called, plasticity it's a fancy word for flexibility. We've seen flexibility in whether or not fertilized larva can become a queen or not based on nutrition, depending on how old the workers decide to feed the larva royal jelly. The same thing is seen in the transition between being a nurse and being a forager. There's flexibility. If you, as an experimenter, take different cohorts of ages of bees and keep them from doing what they typically do, they will be flexible enough that they can start doing other tasks. And that is what you can see. If you have an overexpression of old workers, like 30 to 55-year-old workers, they will produce brood food, which typically is produced by the young workers, the nurses. But you can still kind of force bees into doing stuff that they typically wouldn't do under normal circumstances. So those overaged workers will show no true signs of senescence. Again, if you're precluding them from foraging, they will still continue to act as kind of immature. They're just very immature bees, and they will live longer that way. And as soon as you force them to forage, they will immediately have high mortality, which shows us that it's so metabolically demanding to be a honeybee forager. If you have young foragers on the other end, and you kind of force the worker force to start foraging early, because you might be removing the nurses from a colony, for example, and the old bees from the colony, immediately you start shifting the foraging age so that they start foraging early by removing those nurses. You increase consumption in resources, and you change the lifespan of those bees under a different dietary control. So you can do a lot of things within the colony to change the age composition and the onset of foraging, the onset of juvenile hormone production, the stoppage of the vitellogenin production, and changing kind of the overall age of the worker force. What are some of the advantages to aging in honeybees? So immunological stress leads to foraging and underfeeding, because the nurses are no longer feeding the bees. There's like, you're old enough to go fetch for yourself. I'm not going to feed you anymore. Will lead bees to starting the foraging career. And as I've mentioned, foragers will die in the field from exhaustion, basically, from just being too harsh on their bodies, going out all the time. So there are these evolutionary mechanisms for protecting the colony from sickly individuals, because they will just get very, not sick necessarily, but just weak, and their immune system is compromised, and then the foragers will just die in the field. And so the older bees are performing the most dangerous taxing tasks that ultimately take their lives away from them. So I wanted to mention things about juvenile hormone and vitellogenin that we might not know as beekeepers. There are key hormones that will maintain or dictate not only when nurses turn into foragers, but also what other factors are affecting aging in honeybees. Now I want to dwell into the other topic that really has not a whole lot in common, but some aspects of foraging that's really cool, really cool. Another aspect of the biology of the colony. So let's do some bee math and try to learn about what proportion of the colony's worker force will do one or other foraging task, etc. About 25% of a colony's worker population gets involved in the foraging tasks. Most foragers will collect food within 1 kilometer from the hive, although we know that they can go as far as 5 kilometers away or so, and they will dwell into further distances if there is no good forage nearby, because it's more expensive metabolically to go further distances. So if they can find a good patch of food nearby, they will go for that one and try to exploit that one as much as possible. Up to 14 kilometers has been the recorded distance that a colony can go and send some foragers. And again, it can be because there is no other food available nearby that the colony has to go that far, but it will be very, very energetically costly. So hopefully the nectar in those flowers that are so far away will have high enough sugar concentration that it's worth their while to go that far away. Since we know that the floral resources are patchy and vary in their quality, they're going to have to go for the best available food resource at any given time. If we have this multi-floral area with very diverse food resources, of course the bees are going to be not only getting a more nutritious diet, but they will be getting amino acids that are lacking in some species over others, and they will have a better variety to choose from. So let's do some bee math. So let's do some feral colony bee math. Not the one, I mean, a lot of these things are similar in regular managed colonies, but if you want to look into the true biology of honeybees, you want to do them with feral honeybees. So a colony will have anywhere from 10,000 to 40,000 bees. Just a rough estimate for you to remember, because for instance in the United States we buy bees in packages when we start colonies, and they sell the packages by the pound of bees. And so there's about 7,700 bees per kilogram. So a colony overall, depending on the number of bees, has about 1.2 to 5 kilograms of biomass. The workers and all of their food contents, etc. There is an estimate that a colony will rear about 150,000 workers in 1 year. It's a huge production of workers, 150,000 workers per year per colony. One pollen load from a bee roughly weighs 15 milligrams. One nectar load weighs about 40 milligrams, and it has about 40% sugar in it, whereas honey will have about 80% sugar in it. So we'll talk about evaporative cooling and the honey processing numbers later. But imagine that, 40 milligrams of nectar in one load. In the winter, colonies will consume about 1 kilogram of pollen and 25 kilograms of honey. Now we're talking orders of magnitude. Imagine that each load is only 15 milligrams, but here you have 1,000 grams of pollen collected by, used up by a colony in the winter. And in the summer, because we have the brood production, that number is multiplied by 20 kilograms of pollen used up by a hive in the summer, and 35 kilograms of honey in the summer. So, in order for the colony to be able to do all this math and keep up with all the demand of resources, a colony will have to do roughly, on average, 1.3 million foraging trips for pollen and 3 million foraging trips for nectar. 3 million foraging trips for nectar. Can you imagine that? And a production of about roughly 150,000 workers per colony. Where do foragers go when they go out and forage? Here's a map. This is a typical map of foraging that has been recorded by Tom Seeley in the areas up in Cranberry Lake in the northeastern United States. So he had his hives here, and he wanted to see how far colonies could go from this central location. And of course, there were colonies that were foraging at 1,000 meters away, 2,000 meters, 3,200, etc. But the foraging environment, if you look at radially from where the location of the hives is, it can be anywhere from 13 to 600 kilometers square. It's a huge, huge area, especially if you think about the bees that when in times of dearth can be collecting as far as 14 kilometers away from the hive. The patches of floral resources are commonly small, so they can be as small as 100 meters square. So a colony has about a 70% chance of finding a resource of this source, the 100 meters square, within 13 kilometers square, which is in the low range of this foraging range. So the number will drop off from there, going higher depending on whether they have these patches of the right size or not. In beekeeping, for those of you who are beginners, we have the words honey flow. So honey flow is the period of high nectar collection. And in places perhaps where you have monocultures in a precise time of the year, then you will be able to say with some confidence that your honey is coming mostly from the nectar that is turning into honey from your colony at that particular time in that location can be sourced back to that floral source. But in areas where there's a lot of wildflowers and you don't really live nearby a monoculture, you can only say that your hives have wildflower, multi-floral sources. And the times of dearth are the periods of low nectar collection. This, I just wanted to show the type, this is a very common Texas plant. It's called, does any of you know what this is? Very common where we are in central Texas. Horse mint. If you ever heard of horse mint honey, that's what it is. And it's huge in central Texas. And then we have very small, what is this? Huh? Yeah, some goldenrod. But the type of goldenrod that I was used to in northeastern United States was really long, tall, and very bright yellow with a lot of different clumps of yellow flowers. The one in Texas is very short, and you can almost miss it. So those are when we have the honey flows, these are dandelions. And we have a different times of honey flows for these three particular crops in central Texas. Well, let's talk about foragers and what they go for typically when they're near specific flowers or for nectar or pollen. Foragers tend to collect only one single resource throughout their lives. They like specificity. They don't like switching back and forth. Typically, you will find bees in a colony that will prefer to forage for nectar throughout their lives. And you will typically find those workers only collecting nectar. And the same goes for pollen. So bees that tend to collect pollen will go for pollen most of their lives, if they can. If it's rough out there and there's not a lot of food, they might switch. But if they find pollen, they will specialize in pollen collection. The same goes for water, and the same goes for propolis, by the way. There's some incidental cross-collection, as I said, but it's mostly because of the inability to find the resource that they are most specifically attached to. So if there's limited demand for the resource, then they remain in the nest unemployed. They prefer to stay unemployed than going out and foraging for something else. Sounds a lot like some people that prefer to file for unemployment than go and perform a different profession if they can't find a job in their profession. So as I said, bees tend to visit only one species over an extended period. So not only are they focused on foraging, for instance, in just pollen, but they will try to go for just one type of pollen as long as that pollen is available. So they're not going to going like, I feel like dandelion pollen today. I love the taste of some horse mint pollen, so I'm going to switch today for horse mint. Typically, they don't do that. They will stay within that source. That's what we call in pollination biology flower constancy, until the resource profitability drops. So about 95 to 98% of the pollen returned to the nest by a single forager is from one species. That's why with some very high degree of confidence, if you take out the pollen basket or of the pollen ball off of the pollen basket from a forager, you're almost guaranteed that there's only one pollen type in that ball. And that's also how people analyze pollen pellets from bees. They just segregate them by color, for example, and then under the microscope, they will find that each pollen ball is almost 95 to 98% the same pollen species. If a bee is forced, say artificially or because there's pollen dearth out in the field or flower dearth, if they're forced to switch plants frequently, they will take longer in handling times. Again, this genetic diversity in the hive has allowed for these lines of bees that are specialized in a particular task, foraging task, and they take shorter time if they're a pollen forager to process pollen than if they have to switch to handling nectar. And over time, their skill improves just like it does with us, so practice makes perfect. And the odor recognition of rewarded plants is immediate but decays rapidly without repeated reward. So flowers have learned that they have to offer this floral scent and reward to the bees for them to remember to go back to those same sources over and over. How about acquisition of resources? Colonies take advantage of many, many temporally variable resources, and then the foragers that find that suitable location with enough food will will try to recruit other bees to those sites only after about 20 minutes of them locating the site. They're not going to do it right away, 5 minutes into having found the location, but they will take their time to explore that location a couple of times before they start recruiting. But within 20 minutes, they will start producing the waggle dance or the round dance after discovery to start recruiting to that very profitable source. About 10 to 20 bees will be recruited every hour after a forager found a very profitable resource. That leads to a very rapid acquisition of resources. I think I'm kind of preaching to the choir that honeybees try to make good decisions in foraging as long as they have availability of resources. This is a typical experiment where you change the sucrose profitability of an artificial feeder that you put in front of colonies that you train foragers to in order to test whether bees prefer the high sucrose concentration food sources compared to the low sucrose concentration ones. For example, all of these typical diagrams these are studies by Tom Seeley, but a lot of people use this kind of diagram to show recruitment. You have a hive, it looks like a little skep hive. Then you have your feeder, because typically you have it in a tripod or in a little stand. And you train bees to maybe 100, 200 meters away from the hive in opposite directions. And then you give a feeder either, for instance, 2.5 molar concentration of sugar. That's really, really sweet. Typically, you don't find that very high concentration in nectar. Or you give it a low reward sucrose concentration of 1 molar. And you start counting the number of bees that get recruited as soon as you start training your bees and you paint them, just like I showed yesterday, painting foragers to the homes, the nest boxes. Once you start painting the foragers, you start getting all of these unpainted recruits, because then you also go back to the hive and look at the painted foragers, and you see them performing the waggle dance, recruiting bees to those sites. And you immediately see an increase in each dot of these is the number of recruits, so 1 recruit, that you see increased number of recruits to the feeder that has the highest sucrose concentration. If in the afternoon you switch the sucrose concentration so that now the other feeder, say the north feeder, is the one that has the high reward compared to the south feeder, the colony knows, and they switch the preference of the feeder, and they will start recruiting at higher rates to the feeders that have the highest concentration. And the same happens when you do it on a different day. You can switch back and forth, and you will always see highest recruitment to the feeders that have the highest food reward. So colonies can discriminate the concentration of sugar in nectar based on as little as 0.2 moles per liter. So very low sensitivity ratio. And they collect about 3 milligrams of sucrose per nectar load, changing or altering the foraging patterns to match the changing resource profitability. Sometimes some flowers will flower or provide nectar in the afternoon, some in the morning, so you will see highest recruitment in the morning for the flowers that have the nectar in the morning, and then you see them increasing the recruitment in the afternoon to the other species that offer nectar in the afternoon. One very important group of bees is the scouts. The scouts, as the name indicates, are the bees that seek for new resources. I talked a lot about nest site scouts yesterday, but most of the scouts that we typically talk about are the food scouts, because those are the ones that will go and look for new food sources. They are about 10% of all the unemployed foragers. And the number of scouts will depend on the amount of dancing occurring already. So there are positive feedback loops in honeybees, but there's also negative feedback loops in honeybees that maintain that homeostasis of the colony. If there's a lot of dancing going on, you can assume, or the bees, I guess, are assuming, using anthropomorphic terms, that there's enough food out there because there's a lot of recruitment. So the scouts don't need to go out and look for food. So that's a negative feedback loop, because the higher the resource foraging or waggle dancing, the lower the need for new food sources. Once the number of those waggle dances goes down and there's not a lot of recruitment, then scouts get activated and they go out and look for food. Most of the bees, it's cheaper for a bee to just follow a dance and go to a food that has already been found. Scouting is expensive because you're going out using, contents of your all the nectar that you need to load up with to just start going looking for food. Whereas, so it's cheaper to go and look for food that's already there. You just need to get to the place, but you know that there will be food there, than going out and just looking without knowing if they'll find anything. Interestingly, the probability of performing the waggle dance, here you see a picture of a waggle dancer this is a really cool picture because you can see the bee vibrating, so performing the waggle run, and then all the followers of the dance that are getting all of that information about it. The probability of dancing is based on resource profitability and colony condition. The fewer resources available, the more likely that bees are going to perform the waggle dance to sources that have very low sucrose profitability. The probability of dancing is also modified by the length of time that will take for a food processor to unload the crop. If it's taking a long time for a waggle dancer or a food collector to come back and get her food unloaded, it will take a lot of time for that food to be processed. The likelihood of performing waggle dances goes down. So it's all, again, homeostasis. What signal is used by bees to get their crop unloaded if they're taking too long to get unloaded? What signal is used by waggle dancers or foragers that are coming back and they have a full crop that they need to get unloaded and they haven't gotten it for a little bit?
Don't they get nudged in
the
side? Don't they get nudged in the side?
Yes, but there's a very peculiar behavior that I encourage you to go and look in your hives because it's fun to watch. You can Google it so you can see it on YouTube. No? Oh my gosh, the Tremble dance. They look like they're having an epileptic seizure. When they come back, I'm sorry that I don't have a video of it, but if a bee comes back and it's taking a long time to get unloaded because all the unloaders are busy unloading food, this happens during honey flows, then the bee gets kind of upset. It's like, come on, unload it. And she starts going, da, 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 da. And you think that she's having a seizure, but it's a really cool behavior that tells others, like, unload me. That's the message. So there's a lot of signals that will either inhibit or excite bees to do stuff. During dance duration is based on nectar concentration, distance, and overall profitability. We know that, right? The closer the food, the better. The sweeter the food, the better. The more flowers in a patch, the better. So the bees have to make this decision of whether to dance or not based on colony needs, the amount of dancing already being performed, the time that it's taking them to get unloaded, etc. So they have to make this very convoluted decision. So here's a bee that is going to decide. She comes back with a load of food, and she can decide whether to perform the waggle dance or not, or the round dance, to recruit other bees to that site. And so she has this very important decision, to dance or not to dance. And so here we have some of the variables that affect her decision. The distance of the food from the hive, the concentration of nectar of sugar in the nectar, the abundance of nectar, etc. And then in the forager context variables, you have the colony's nectar influx, so how much nectar is already coming in, the weather, because they don't like to go foraging when it's pouring out, even if there's food out, the time of day, they prefer to do it during, you know, the middle of the day rather than late afternoon or so. So both the profitability of the nectar source and the threshold of the dance response will help a worker make the decision of dancing or not to recruit other bees to that. So the forage site and context will vary over time, and that bee will take into account the profitability of the nectar source and what the dance threshold is at a given time, so the amount of need of the colony to decide whether or not to dance. And then she will dance based on those factors. So if she dances, she will adjust the duration of the dance, the number of total waggle circuits based on how profitable that site is. So I don't want to talk about the waggle dance and how it's done, but I want to mention that the profitability of the food source that a bee is waggling for is correlated with the duration of the waggle circuit. So how many H-shaped figures a bee will perform in a given time period. If she doesn't do as many, like say 10, for example, compared to 100, because you've heard of the waggle dancers that perform marathon dancers, that is telling the colony that the food is really high in profitability. So here you can start thinking about it theoretically. You can have the profitability of a nectar source that can be either low, medium, or high. And here you have the duration, or the total number of waggle circuits that can be a short duration or long duration based on the nectar influx, which is either low or high. And so the bee will adjust the duration of her dance based on the profitability of that resource and how much nectar influx there is. So very complicated stuff. You can think of a bee just getting some food from a source and think that that's it, but really she's making all of these very, very convoluted decisions based on all the positive and negative feedback loops that are happening inside the colony. This is also just showing that bees will increase the feeding to a specific site based on whether it has the high or the low sucrose concentration. So I think I kind of mentioned that already. So colonies collect nectar more or less continuously, and daily foraging is regulated, amongst other things, by the DVAV. So remember I mentioned the DVAV yesterday during swarming. It's really not involved in swarming so much. It is involved in helping to activate queen behavior, so she starts laying more eggs and starts losing weight, but it's really not done as a signal that triggers swarm departure. I told you that it's more of a get active kind of behavior, and the DVAV, the dorsal ventral abdominal vibration, or we call it the shaking signal, it has that general message of get active. That bee that is performing the DVAV behavior on another one is telling her to get active, and it happens, it's highly done in the morning when foraging is getting activated. So it's almost like a wake up, it's morning time, rise and shine, let's go and look for food. So that DVAF is one of those positive excitatory behaviors that will aid in the onset of foraging during the day. So this is the Tremble dance. Really cool behavior. The bee quivers and runs, shaking back and forth with forelegs held aloft. They can perform it up to an hour, for up to an hour. And so you see, if you count, if you look at the behavior or the performance of waggle dances, the number of waggle dances, if they start dropping, the waggle dances will start to drop if you see an increase in the Tremble dance, because they're not getting fed. Unloaded. So the Tremble dancers are fed up with the fact that they're not getting unloaded, and that production of the Tremble dance will tell the waggle dancers there's too much dancing going on, so stop doing it. Okay, I want to move on to the other study, because otherwise, I want to talk about water needs. So not only colonies foragers are recruiting for pollen and nectar, they're also collecting water. Water is needed in the dilution of brood food, because brood food is about 70 to 80% water. Honey also has water, about 16 to 18%. But water is also collected for thermoregulation, to keep the brood nest temperature for about 33 to 36 degrees Celsius. I told you that each kind of line of bees specializes in a collection of specific resources, and in a colony, there's only about 1% of the foragers that specialize in water collection. And they tend to just collect water. They don't go for the other pollen nectar. Middle-aged bees can be water unloaders if needed. And water collection, amongst other things for food, concentration of water, is done for evaporative cooling, which is a really cool behavior that you will probably see in the summer, where you see bees fanning. But before fanning, those water collectors will bring back water droplets to the hive and start fanning the water next to them, the water droplet, to lower the inside temperature of the colony. It's basically a mist that helps lower the inside temperature. So they spread the water over the comb face, they fan it, and that helps create this cooler mist that lowers the inside temperature. And bearding is also done when the colony is trying to thermoregulate. So you will see the water foragers performing the evaporative cooling behavior, and then the rest of the foragers will be outside creating those beards that I talked about yesterday that are so important in thermoregulation. Finally, they will search for propolis, which is that resinous substance that's collected by propolis foragers, used to fill crevices, seal the openings, and as a form of self-medication, because it has antimicrobial properties. So feral colonies will have an envelope of propolis all around the inside lining of the cavity, and that helps maintain the colony free of bacteria. And a lot of work on this self-medication by honeybees is done by Dr. Marla Spivak, S-P-I-V-A-K, if you're interested in looking into that, at the University of Minnesota in the United States. She's not only just a wonderful speaker, but she's doing really cool stuff with propolis and the antibacterial properties of propolis in honeybees. Humans have kind of known this for a while, and we collect propolis, or we harvest propolis for the cosmetics or medicinal industries. So I just wanted to mention all the things that bees can collect and how they coordinate the foraging of all of these resources. So for the last 10 minutes or so, I want to talk about Nosema, because I promised that I would talk about it. Completely different topic that has nothing to do with the previous two, but I wanted to talk about our Nosema study because it's really cool stuff. Someone asked about the presence of either Nosema species, so I will go into this. So we just actually, this week, we got the proofs of a paper that's going to come out in Apidology on the prevalence of Nosema in feral honeybees in South Texas. So this is just coming out soon. So if you want a copy of the paper, you can email me. And again, I have business cards, so you can have my email. So I'll talk a bit about Nosemosis. Microsporidia is a group of parasitic fungi that have over 200 species of Nosema. So disease by Nosema is called Nosemosis. It's one of the most prevalent diseases in adult honeybees. It's caused by two species nowadays, Nosema apis, which was the original species affecting Apis mellifera and Apis mellifera, and then Nosema cerana, which switched hosts. It used to just occur in Apis cerana, and now it occurs in Apis mellifera as well. Infection with Nosema begins when a honeybee consumes the spores of this microsporidian, usually when she is cleaning bees, removing spore-infested feces from the comb to get these cells ready for the next the next egg that's going to develop in that cell. Then the spores will travel through the bee's esophagus, through the crop, and get ultimately into the meat gut. When the spore germinates, I don't know if you can see it in this picture, it shoots a long tubular filament, it's called the polar filament, that penetrates the midgut cells of the bee. And once the spore shoots the polar filament and it attaches to the epithelial cell, it can start hijacking the epithelial cells to grow and multiply, forming new spores. And then that's the way that the spores germinate in the midgut and infect more and more cells. So I know that Dr. Delaney talked about nosemosis a little bit. I don't know if she did in the later lecture, but in the first one about diseases, so I'll go over some of these common pictures in more detail. So the midgut of infected workers with Nosema apis, remember apis, appears white and distended, and their respective colonies exhibit the very common fecal streaking on hive bodies and the comb. Very infectious way of transmitting these spores because they're in the feces. So the bees can't. Typically, bees are really clean. They will use the restroom outside of the hive to keep everything clean. But dysentery and diarrhea do not allow the bees to wait until they come out, and so you see the dysentery on the outside. However, infection by Nosema cerana does not show these symptoms, and that's why it's so difficult to diagnose a colony with Nosema cerana. That's one of the big problems, is that there aren't these obvious symptoms. Colonies infected with one or both species of Nosema have shown reductions in the population of workers, the reduction in honey production, and colony mortality associated with digestive disorders and shortened lifespan. So workers that are infected with Nosema have shorter lifespan. And as Dr. Delaney said, a lot of this is happening because they're malnourished. Even if they're getting food, because the epithelial cells are compromised with the spores, they're leaking out all of the nutrients and they're not getting the food that they need to perform all of their regular digestive and metabolic processes. The spores, as she also mentioned, are very difficult to recognize under the microscope. So Nosema apis spores are typically a little chubbier and shorter, whereas Cerani spores are a little longer and thinner, but it's difficult to have a reliable diagnosis of which spores a colony is infected with just using microscopy. So nowadays, most of the Nosema cerana discoveries are done using genetic markers, which can be quite expensive to do. Nosema apis in the US was believed Nosema apis generally was believed to be the only species to infect European honeybees. Until 1996, and now we believe that it's much earlier than that, maybe the 1980s, Nosema cerana was found in managed colonies and is now present in all colonies where beekeeping is performed. However, very few actually no, quantitative studies, have looked at the presence of Nosema, apis, and Serrana. They have done a couple of studies looking at just Nosema apis because they were doing it microscopically back when there was only apis around with Africanized honeybees. And remember I showed this map of the Africanization of honeybees in the United States. So this, to date, is the first study that has ever been done on Nosema in Africanized honeybees with both species. So our question was, what have been the levels of Nosema in feral honeybee colonies in South Texas over the last 20 years? So our study site is the Welder Wildlife Refuge in South Texas. So this is the huge state of Texas, and here's the Welder Wildlife Refuge, which is a place where you can conduct research of endemic species and naturally living species in a very large area. And so there's a very nice 7,800-acre area where you can do all kinds of studies. 80% of it is covered in chaparral and mesquite, very thorny, short shrubs, and it's very, very humid, especially in the summer. Interestingly, for the last 20 years, they've been able to do a mapping. I don't know if you can see the green dots, but around the Welder Wildlife Refuge for between 1991 and 2001, they were able to locate post oaks and other trees that contained feral honeybees in them. Those were the first collections in 91 to 2001. And then in 2013, we went back to the Welder, 12 years after the latest collection, to look for bees in trees. So they found 112 bee trees from 91 to 2001. 85% of those lived in post oaks. And then we went back in 2013, sampled 90 of those trees because some of them were dead or they were really difficult to get to. And we only found bees in 27 of them. That's a really cool study about landscape ecology as well. So we collected bees and then we sacrificed them, so we would go up to the tree in the post oak and collect bees from those trees. These are some pictures of the collection. One guy in particular was very eager to just climb up trees that were 20 meters up without any ropes or anything, with his bee suit on and his net, and he would collect the bees for us. So very important guy, because otherwise it would have been difficult to collect bees from some of these trees. And then we estimated, first of all, under the microscope, the average number of spores in these colonies. Then we wanted to see if there was any correlation at all of Nosema levels based on the climate changes over time, the precipitation and average temperature in July, which is the month when we did all of the sampling. So we performed statistical tests to determine if the mean number of spores varied across years. And then we wanted to see if temperature and rainfall had any effect on the average copy number of Nosema species. So all samples were frozen until the molecular analysis was done, which was done by Dr. Brenna Traver, who is at Penn State Schuylkill. It's a very weird name, a small campus of Penn State in Pennsylvania. And we extracted genomic DNA, so not mitochondrial, like the one that you use to decide whether a bee is Africanized or not. We used nuclear or genetic DNA analysis. From each colony, we sampled 10 bees. And then we performed what is known as quantitative PCR or real-time PCR using a standard method to analyze the number of DNA copy numbers, we call them, so it will tell you how many reads of either Nosema, apis, or Serrana we were able to get from each bee sample. So as far as the number of spores per bee from worker samples that tested positive for Nosema, we analyzed bees from 91 through 2013. We analyzed almost 500 bees, but we only found Nosema spores in 28. So very low levels of Nosema overall, just doing the microscopy work. Here we have the mean spore count per bee that ranged from a few hundred to a few million. We did not find any effect of temperature or rainfall. So here we have in red, we have the precipitation or rainfall. Here, precipitation, so in millimeters in the month of July. And here we have mean temperature in July. It basically didn't really change very much. It was about 35 degrees Celsius every July. We did not find a difference in the expression of Nosema based on precipitation or temperature. The levels of Nosema apis steadily decreased since 1994 and disappeared in 1998. In the red here, this is the number of colonies analyzed. In the red is the proportion of samples that showed infection with Nosema apis. So about 30%, then it dropped to about 5%, then it went up to 30% again, and then it fully disappeared. Nosema apis was not seen after 1995. In contrast, Nosema serrana has been going up steadily and now is present in about 95% of the samples in that population. If you look at those copy numbers that I told you about, the kind of the level of DNA expression based on that real-time PCR genetic test, in the red we have the levels of Nosema serrana. In the gray we have the levels of Nosema apis over time since 1991 to 2013. You can see here that the levels of Nosema apis were, kind of steady for the first 5 years, but they dropped in 1995. And now we have very, very high levels of Nosema serrana. So it really looks like the same pattern that we've seen in the United States and other parts of, in other continents, as far as the interaction between Nosema apis and Nosema serrana. So the levels of Nosema overall in feral colonies in this part of the country have changed over the last 20 years with very interesting population dynamics. The percentage of colonies exhibiting Nosema serrana have increased from 18% to 86% in 2013, while the percentage of colonies with Nosema apis decreased from 33% in 1991 to 0 after 1995. So, just like many studies have shown or have argued, Nosema serrana seems to be displacing or have displaced in this population Nosema apis, just like it has been seen in other parts of the world. But definitely more research needs to be done on the levels of Nosema serrana and apis, especially in feral honeybee colonies, because even though we saw that infected colonies had high levels of Nosema serrana, overall, there's not a very high incidence of Nosema in feral bees, which is not the case in a lot of managed colonies across Europe and the United States. In fact, in Spain, it's been proposed that Nosema serrana infection has a lot to do, as a key player in colony declines in that country. So I just want to thank the people that collected bees in 2013 at the Waggle Wildlife Refuge. I really want to encourage you to go to our to our Facebook page if you want to just drop us a line and ask any questions you want regarding our research or anything related to honeybee research. I would like to thank the Blacksland family for their generosity in providing the funds for this lecture. Simon again for his invitation to come to the UK. I really encourage you to email me or take any note cards from me if you want, so we can talk further after this. But it has been a wonderful experience to be here sharing some of my knowledge with honeybees in the UK, and I really thank you for your attention and generosity.
I had a question about this marathon waggle dance, and trying to understand why the bee would continue to do that. I understand because of the high productivity of the nectar flow, but presumably fairly soon on in the dance, the bees will leave to go for that source. So why do they continue on? Is it to instruct the bees returning from other sources?
So he's asking about marathon dancing, and why would bees perform such long marathons of dancing Because
Because
presumably,
the bees have left.
The bees have left.
Will watch
so
them leave, and they'll be using up energy using that marathon dance. So I don't
yes,
understand
so
why they do that.
dancers will, if they're performing marathon dancers, they will stop a little bit to fuel up again and continue dancing. But they get really excited because the food has to be superb for them to perform these dances. So followers will follow the marathon dancer, then decide, because they also have to make the decision of to follow or not to follow, and then to go and look or not go. But if the dancer is really eager, then they have a higher likelihood to go out and forage. Then they will go. But then she can stop a little bit and have some food and then continue dancing, and then recruit a completely different group of foragers, of followers. And what's really cool about this is that if a marathon dancer performs the waggle dance for, say, 30 minutes, even up to an hour, the direction or the angle of the waggle run will change accordingly. Because remember that the direction of the waggle run correlates with the direction to the food source. She has that internal clock that tells her about what time it is, and then she will start changing the angle of the waggle run so that she is telling the correct information to the followers. It's really cool behavior.
Does the proximity to the hive matter when you're proposing to put a food source for bees, an improved food source? Is there a distance too close that you must avoid?
Does the proximity to the food source influence the decision of whether to dance or not? And is there a place that's too close to the hive?
Yeah.
No.
No.
There's no place too close. The closer, the better, right? But bees will perform the different flavor of the waggle dance known as the round dance if the food is about 50 to 80 meters from the hive. Then they will transition to producing the full-on waggle dance about 80, 100 meters away from the hive. But the waggle dance, and we believe that this is an evolutionary strategy, is not 100% accurate. There's a little bit of error in the angle at which that waggle run is produced. And we don't believe, or studies don't believe, that this error is because they're making this mistake that they can't really aim well or have the correct angle. Is that it's giving bees a little bit of breath to go and look for anything that might be close to that angle, because perhaps there's a patch of that same flower a couple of degrees to the left or the right.
I mean, say 5 meters from the hive,
Oh, no,
or
they
50
will go.
meters.
Yeah, they
it
will go close to the hive, absolutely. I mean, if they're lucky enough to have something nearby, they will go for that. But it depends on whether there's nectar available. So it's not always that if you see the flowers, they're going. It also does not say that if you're, say, in your garden, you have flowering flowers and you have bees on your flowers, that those bees are coming from your hive. They might not be. They might be from other colonies, and your bees happen to be somewhere else. There's also, there's where I said flower constancy is not just daily. So a bee that was really successful at obtaining food 100 meters from a hive, she will try to go the next day and see if that resource is still available. So she will still go there instead of trying to scout for something closer.
Along that line of going, if they see the specific flower that they travel for, if I've taken a group of bees out of their natural environment and moved the more than 3 miles distance, say, for instance, I've taken my hive from Wales and moved it up to the Midlands, I specifically bought flowers that I knew that the bees would enjoy for the pollen and the nectar. But sort of what this gentleman was saying, I've put these right underneath the hives and the bees are ignoring them.
Well, no, I guess they need to still go out, right? And kind of say, where am I? So putting them right on their nose might be too close, yes.
I've
So
even
you
picked
want to
up
do
bees
that.
and throwing them onto it. Look, it's food.
Well, that's how we train bees. If you have a feeder at the entrance, sometimes you put a stick full of honey or sugar so that the bees actually walk on that bridge onto the feeder right on their nose and then start moving it back so that they can get trained to it. But in reality, you know how sometimes bees leave the hive, they're kind of like, and then they start looking. So too close to the hive might not be the best
But if that's a food
strategy.
source that they wouldn't normally forage, because obviously I bought ling, which is from Scotland, and these are bees that have come from Wales, and they're being brought up to the Midlands. So there's a differentiation of what they would normally forage. Would the newer bees coming out of that hive learn that that's a more forageable source?
It depends. So will the bees that, if she placed these bees in a new location and put these flowers that you know bees tend to go for, it will depend on whether they've already found, because you have all these scouts, right, in the hive. If you find a scout that just happens to prefer to go further away and found something profitable there, they might start recruiting to something that is further away than your flowers in your garden. So it all depends on when they were found and how much recruitment there will be to it. Thank you. Thank you.